So if you go into the pathway folder under MEPM pathways and other, you'll see this diagram here. And of course you will recognize this pathway from the one we just drew, which if we go back here, put them next to each other. So this is what we drew. Yep. This is what we attempted to draw, I should say. And I'm going to point out what we've done to it to turn it into a computational model. So first of all, we have interferon beta. So before we didn't have any interferon beta because it was just a picture. So now what I've done is essentially, so in our, let me go back to our palette. You'll see these guys here. So this is a symbol that we use to say this is the start of the process. Okay? And so what we can do here is we can take this here and we can run an edge from it here. And we ran an edge from it. And then we click on that edge, properties, and we can put in tokens. Okay? So just think, as I mentioned, tokens are like how much of this thing is there. Okay? The more I put in, the more we're assuming that that interferon is present. So we could run simulations where we assume there's 100 tokens, or we could run simulations where there's 1,000 tokens. We're assuming that this represents the amount. So let's say 100. Okay? Okay. Now, in this diagram, these are working as transitions. So all of those little process nodes that we added to the diagram are actually transitions. So these things are different in the way that BioLayout will interpret them. These will be interpreted as transitions. These will be interpreted as places. So our notation system has now been mapped onto BioLayout so that there's a parser within BioLayout that will recognize the different shapes and recognize them as either places or transitions, and will work with them in that way. So as we fire this down here, we get to this point here. Now, as I mentioned, when you get an interaction between two things coming into a transition, then actually, if we didn't have tokens here, then there would be no receptor. And if there's no receptor, we can't get anything downstream of that receptor. And so we have to assume that these guys here. Likewise, in all of these cases here, if there was no stat1, we can hardly phosphorylase it. If there was no stat2, in fact, and there's stat1, so if all of these things we have to assume are present at the start of the reaction. Right? So I'm just going to take this now, and I'm going to do one other thing. I'm going to get rid of one thing which is quite important. I'm going to get rid of this guy here. And I'll show you. So you can take this diagram, and again, going back to BioLayout, we can drag it in there. And it's going to ask me. So I'm going to say just yes for now. I'll run 1,000. Okay, doesn't really matter. Open simulation. So first of all, I just want to have a look at the diagram itself in BioLayout before we actually come back and do this again. And change the background color to black. Now you see what we've done is we've used those shapes and mapped them onto a 3D type glyph, so that the 2D rounded rectangle has now become a 3D rounded rectangle. The circles have become spheres, and the mapping between what you recognize in YED has been mapped onto here. And also what you can see is this background here. So this is not something that I can click on. It doesn't exist. And it's really just been used so that in the diagram itself, you'll see that extracellular is written within essentially asterisks, and that tells the program to treat this node as a kind of background node and not as a real node. So in fact, one other thing that we can do here is that we can add depth to that. So if I want to say that was 100, apply. I'm going to reload this again. What have I done? I've applied it, but I've not saved it. Save it. I'll reload it again, and you can see now that that has volume. Okay, so by specifying 100 of that, I've given that shape volume, and so if, rather than being sitting on a surface, it's sitting inside a 3D environment. So I've just gone into that shape now. We can see it there, yeah? Okay. So all the usual tricks that you learned yesterday with biolayout still apply. So the model is still there. I'm just going to make it look prettier. Okay, and you can see the shape of that is nicely mapped onto here. Now if we want to just get rid of that background, because it can interfere, we can say shut off the wired styles there, and we've got the diagram as it is. Okay. Now we can see here that we can show all the edge weights, so all those tokens which have got tokens on them are now mapped here. And now we can do exactly what we did before with the big lattice, but now on our favorite diagram. And we're going to run this now, run SPN, run the simulation, and open up simulation control. And now we can do exactly what we did previously. Again, I'm going to drop this value down here. And we can start this animation so that now we can say, well, how would this might work in reality? Now you saw something quickly happen there, and then it stopped. The reason that it stopped, let me just stop this a second, let me just make that bigger. So I'm going to make our nodes bigger. We're dealing with a smaller diagram, so we can deal with the smaller nodes. Start animation. See this guy? This is our inhibitor. This is our SOX3 that we had being produced here, and it's come back here and it's stuck there. And the reason it's stuck there is because we've added tokens in, and this inhibitory edge does not lose tokens. It's just the nature of an inhibitory edge. You don't lose tokens through an inhibitory edge. So tokens have accumulated here, and what we will see is that if I click over this now, let me just stop that animation. It's not clear, but you can see I've got tokens up, and then it's fixed to reached a maximum, and it's not going anymore. Okay, so it's accumulated tokens, and because the inhibitor is there all the time, it's blocked any more flow through that network. So when we build inhibitor edges, we need to sort of begin to think about them in a different way. Okay, so we need to add, so we have this node here called a sync node, and a sync node is where we actually lose. So the sync is generally used to show something being gained from a system or lost from a system. And in this case, what we're showing is that this guy is being lost for the system. Now, if we add this now, then tokens are going to be lost from this node. So it's kind of like building the concept of a half-life into a protein. So a protein accumulates, but then it's destroyed. And when it gets destroyed, then it disappears from the system, and therefore there's no longer an inhibition on the autophosphorylation, and the signaling pathway can open up again. So having made that change, now let's see how it works here. So I'm just going to save it, come back here, open that up again, run the simulation, and what you'll see now, I hope, is that this has been turned into an oscillating system. Yep. So just that making that small change and introducing the idea that the inhibitor is destroyed means that when it gets destroyed, it opens up, the system is now allowed to flow, but obviously one of the consequences of allowing tokens in the system is you produce more of an inhibitor. The inhibitor comes back again, blocks the pathway off, and you get this simulation here. So now if I stop that here and look at any given place down here, we can see we've got a kind of oscillating system going down here. Probably easier to see if I run slightly less runs. So if I take that down to 200 perhaps, run the simulation, very quick now, very small diagram. And let me just stop that. I need to just check that back again. Okay. Very quick. You might want to slow that down. But you can see now, from whichever angle you look at it, that this is now oscillating in a way. And if we go back here, you can see that nicely this system is oscillated. Okay? Does that make sense? So the other thing that we might just want to try and do, and this is just playing around with concepts of how stochastic, how random is a biological system. So in this model mode here, as I say, we are taking 50% of the tokens forward. I'm now going to change this to a uniform distribution, run that simulation again, and now if I look at it, you can see how more random it becomes. Just changing the assumptions behind how random the flow of tokens is completely changes the dynamics of the system, and suddenly that nice clean oscillator has become a far more noisy affair that we can begin to see here. Everything is oscillating slightly. Let me just stop. Again, going back to changing that to 150. Well, you see, you can begin to see that. Again, if I take this back now, and now I reduce this to only 5 number of runs, so we're going to only average 5 runs here, and run it again, you'll see that this almost stops, well, it becomes a very random 250. Yeah, so even the output of these genes is quite different because, again, it's the kind of randomness that we're bringing in. So this is beginning to approximate how we might understand biology working. A series of random molecular interactions between proteins at certain levels, which causes a kind of randomness in the output that we might get. Any questions so far? Are we happy with this? Now, the reality is that when you draw these diagrams, that actually you'll make lots of mistakes. Okay, and the software is very specific about how it must be drawn and how the interaction. So you can start off with a diagram. I should probably start introducing into these courses a diagram that has quite a few mistakes. So if, for instance, I had, let me think, introduce a mistake in here. So I'd forgot to put this point in here, or I'd done something like this. Just okay, so I've now wired that here. So I've broken the Petri net. Okay, I've no longer got this. So I start, save that, open it again. Yes. Well, let me just do 500. Run the simulation, open it up, start. Yep, stop. I think 1500. What's happened? It's got stuck. Because I've broken the system, I no longer have a fully bipartite graph because I've got 2 places stuck together. I've not actually known that. So it's very easy to do when you're drawing. And the one thing that this whole system has brought into us is a kind of a level of accuracy that we never used to have before, because if you don't draw it right, it doesn't work, and those mistakes. So what we see here is there's 1.5. Where is the tokens accumulating? See this? So tokens are constantly coming in, but they've got nowhere to go, because for this place is connected to another place, there's no transition in the middle, and so tokens are just being fed in and accumulating in a linear fashion as they're fed in from the beginning. Make sense? So it brings us level of accuracy in the way that you must draw these diagrams that we never used to have before, because we could never test out whether what we drawn actually worked logically. So it's quite cool.